Thank you very much. First of all, thanks a lot for inviting me today. It's my first time in Korea, and I'm very excited to be here and to meet all of you and to showcase the work we've been doing in my lab for the past years. So rather than talking of a specific aspect of the research we're doing, I try to cover the different research thematics that we are currently conducting in the lab, really to emphasize the challenges we have. So let's start with a bit of background on malaria. So malaria, this is a typical map that you will have in textbooks on malaria. So it's clearly a disease that is mostly affecting sub-Saharan Africa. It's a disease caused by infection with Plasmodium parasites. There's a number of different species infecting humans. It's actually also infecting a wide range of animals, but usually they do not cause any disease in human hosts. It's a parasite that is transmitted by Anopheles mosquitoes, and there's dozens and dozens of different species that can transmit malaria depending on where you are around the world. And in 2023, malaria, and we showed this in the previous talk, was one of the major cause of infectious disease death around the world, with 600,000 people dying from malaria. However, when I present this slide, I'm really just talking about Plasmodium falciparum malaria, which is really the main killer in sub-Saharan Africa. But Plasmodium vivax is actually a different parasite species that has a very different epidemiological profile than Plasmodium falciparum. And as you can see on this map, where you have the incidence of specifically Plasmodium vivax, it's mostly absent from sub-Saharan Africa, although this is debatable and it's probably underestimated. But it's clearly the most geographically widespread outside of Africa, and it's definitely present in the Amazonian forest in South America, in India and Central Asia, of course, all Southeast Asia and Papua New Guinea. And very interesting, so it causes huge number of cases every year, of course. Very interestingly, it's endemic here in Korea, and Korea is now one of the only country in temperate climate that sustain endemic malaria transmission. And the malaria we found here in South Korea, as well as in North Korea, is caused exclusively by Plasmodium vivax parasites. And here on this graph, it's the number of official cases that are reported to the WHO in South Korea over time, and as you can see, it's definitely not going down or even being close to be eliminated. So the reason why there's still malaria in South Korea comes from geopolitical issues, which is very frequent in malaria. It's a disease of poverty, but it's also a disease of war. And so the situation where you have this DMZ between South Korea and North Korea, where there's no vector control that can be done and where there's no intervention, feeds ongoing transmission of malaria in the country. And so we don't have the official numbers for North Korea, but they are most likely way higher than what we see here in South Korea. So Plasmodium vivax has this very specific particularity of being quite resilient to elimination efforts. So this slide shows the number of the proportion of malaria cases in Cambodia in the past 15 years. And what you see is that Plasmodium falciparum cases are really going down with elimination efforts being implemented in the country, while the number and the proportion of Plasmodium vivax cases is really increasing. Today in Cambodia, there's nearly no more Plasmodium falciparum, and Plasmodium vivax is the only parasite remaining that causes malaria infection. And this resilience is due to a number of biological specificities of this parasite, but also technical aspects contribute to this resilience. And mainly, it comes back to the life cycle of this parasite, which is very complex, and I will not go into the detail. But basically, when an infected mosquito bites a human host, those parasites called sporozoites are injected in the bloodstream and very rapidly will infect liver cells, hepatocytes. And in those hepatocytes, they will undergo multiplication, then release parasites in the bloodstream and infect red blood cells. And this is where the human patient is sick, when there is proliferation and multiplication of those parasites in the blood, and then specific stages of those blood stage parasites are transmitted again to the mosquito. In Plasmodium vivax, and not in the other major human malaria parasite, falciparum, a fraction of those injected sporozoites will form what we call hypnozoites, and those hypnozoites are dormant forms that will stay dormant in the liver of the infected patient for weeks, months, or even years after the inoculation by the mosquito itself. And at some point, and for reasons we do not yet understand and we are trying to answer, those hypnozoites will wake up and reactivate and resume the normal development, where they will multiply and then at some point get released in the blood, causing a relapse episode. And so relapses are definitely one of the reasons why it's so difficult to eliminate vivax malaria. It's because after one infection, you get sick multiple times for a very long period. What's really difficult also for elimination is that there is no biomarkers of hypnozoite carriage. We do not know who has those parasites in their liver. Another specificity of Plasmodium vivax is that it does not infect only red blood cell in general, as would falciparum. It infects only reticulocytes, so those very young red blood cells. And this causes very interesting biological questions, but also for us researchers, a major problem, and leads to the absence of in vitro culture system for this parasite. We cannot grow Plasmodium vivax in the lab like we do for Plasmodium falciparum, meaning we cannot have the genetic approaches that we have established for Plasmodium falciparum that can be used for Plasmodium vivax. And this clearly our absence to provide adapted strategies for Plasmodium vivax, as well as those relapses, cause a parasite that is difficult to eliminate. There are, of course, specific needs and gaps in Plasmodium vivax malaria, whether it's on a therapeutic perspective or vaccine perspective, and I will list them here. Therapies for Plasmodium vivax is double. The first component needs a drug to eliminate blood stage parasites, so the one that causes symptom, and those drugs are actually the same than for any other malaria species. Chloroquine was really, and is still, a major antimalarial used. Artemisinin and its derivative are now also coming in. There's a number of drugs actually that works for Plasmodium vivax. And specifically for those drugs, the only challenge that we may face, but this is very complicated to evaluate for Plasmodium vivax, while it's extremely well documented for falciparum, is drug resistance. And this leads back to what the talk we just heard about AMR. The second component that is absolutely needed for achieving a radical cure, so complete elimination of all parasites, is a drug that would target those dormant hypnozoites. And there's only actually two molecules that are on the market. Both are from the same class of compounds, 8-aminoquinolines, and those are really problematic drugs. First, because of toxicity issue, notably in G6PD deficient individuals. G6PD deficiency is the most common enzymopathy in the world, and it's also unsafe for pregnant women. So there's a huge number of individuals that cannot get any of those drugs, so they cannot get rid of their hypnozoites, so they will have relapses. So definitely there is a need for coming up with new drugs here. In terms of vaccine, so we do not have any vaccine against vivax malaria. Falciparum malaria, there's two vaccines that came out and are currently endorsed by the WHO, but the protective efficacy is very low, and I will not talk about it. But for Plasmodium vivax, there is no vaccine. So there is definitely a need for developing vaccine, and there's different strategies. The first one, and this is what's approved for falciparum, is to block the infection of sporozoite into the liver cells. The problem we have to develop such vaccine with Plasmodium vivax, because we cannot culture the parasite, is how can we get parasites to evaluate any candidate that could be used for vaccine development. A different approach would be to have a vaccine that would prevent the infection a vaccine that could be complementary to pre-erythrocytic 1, actually, to block infection of red blood cells. And again, the question here is, where do we get parasites to come up with those vaccine candidates? And the last aspect of vaccine development that is currently being investigated by a number of groups is transmission blocking, but we face again the same question. So there's a number of surrogate models that exist. I won't talk about them. They work a bit, but never enough. And when we try to translate the results obtained from those surrogate models to what's actually happening with Plasmodium vivax, often there's no perfect overlap. So we do need a vivax model for studying and developing whether it's drug or a vaccine. And so the only way that we can do this is really to bring the lab to the parasite. And so this is what we've been doing in Cambodia over the past years, and where we really try to leverage the assets that we have. And what do we have? We have infected patients, and we have live clinical isolates. So we can do rigorous clinical trials, and we can also, and we have developed ex vivo bioassays. All this is because we have quite good infrastructure, equipment, and supplies not that far from those infected patients. Something that we never think about when we're in a country like in Korea or in France is how in low middle income countries, some resources like dry ice or liquid nitrogen are very complicated to get in a regular way. And this is something we've been able to achieve in the country. So Phnom Penh is here, the lab and Institut Pasteur Cambodia is here in the capital. Malaria transmission in the country is very in rural, forested, remote areas, so we have established 3 field sites. The distance is not that far, but the time to go from Phnom Penh to here is nearly 8 hours by car, so it's way too long to have a parasite surviving the transportation. This is slightly closer, so we can do a bit more. What was really important for us is to arrange this network of field site, but also our capacity to actually get a bit of laboratory capacity close to the patient, and a lot of logistic to make sure that when we have a sample coming in or a patient recruited, we can have a turnover in the right time to get either cryopreserved parasite that we perform immediately on site, or shipment for the site that is not that far. Because again, if we wait too long, the parasite dies. It's a parasite we cannot culture for a long time. And so we do a number of things using those parasites, so either fresh or cryopreserved. We can do blood stage culture, so basically the parasite, after it infects the red blood cell, will multiply until it's ready to burst and to infect a new one. So we can do drug response plates and generate IC50 curves, so it's very low throughput and quite isolate dependent, but still we're able to do a number of things, and notably we are seeing that the susceptibility, so these are IC50s to mefloquine, one of the components of the current treatment in Cambodia, has really been decreasing in the past years, and so we are suspecting that there is now emergence of resistance to mefloquine in the country. But we can also, using those kind of assays, validate new anti-malarials, and so this one is interesting for the parasite biology. It's a drug that blocks protein aggregation, and it seems to be quite efficient for not only vivax, but also falciparum and other parasitic disease control. When we do not have the in vitro sample, or ex vivo, I should say, because we cannot culture it long time, we also can work directly with patients. And so this is something that we really try to do in the lab, is to couple clinical studies using infected patients with, as much as we can, either ex vivo assays or genomic analysis. And so this is just an example of what we can do in terms of looking at what the treatment does. So we recruit patients, we give them a treatment, in this case was artemisinin, and we follow them up to see how the treatment works. And what we were able to observe is that the vast majority of patients had their parasite eliminated very fast, but some of them were showing quite a prolonged duration after treatment to really get rid of the parasite, which is, for falciparum malaria, a very clear sign, warning sign of emergence of a resistance. So when we observe this for Plasmodium vivax, because of the difficulty inherent to Plasmodium vivax, we decided to go with a more genomic approach to understand what's happening. And so what we did is we collected a blood sample before treatment and a different blood sample after treatment. So all this is from capillary blood collection, so finger prick. And from as low as 50 microliter of total whole blood, we can generate very robust RNA-seq data, and we can compare the gene expression profile of parasites before and after treatment. And what we showed, and this is something that came out very recently, was that those parasites that are taking long to being eliminated by artemisinin showed a reduced, a downregulation of hemoglobin digestion genes. And so we know that for falciparum, slowing down hemoglobin digestion by the parasite is a way to be protected against artemisinin that is activated by the hem of the hemoglobin digestion. So we believe that what we observe here with those slow-clearing parasites are, I'm not sure we can talk about emerging drug resistant against artemisinin for vivax, but definitely parasites that have intrinsic capacity to sustain a normal environment that should kill them by slowing down their hemoglobin digestion. So a word on the vaccine, preclinical vaccine development. I do not do a clinical evaluation of vaccine efficacy, but because we have this capacity to have parasites that we can grow until they are chysont and ready to burst and infect a new red blood cell, what we do is we actually add uninfected reticulocytes from donors that are not malaria infected. We make them fluorescent and we mix them together. And so you can do this in presence of different IgGs that you want to evaluate the capacity to block this invasion. And using a flow cytometry scoring, we can have this very clear, neat population of fluorescently labeled reticulocytes that are positive for DNA staining, and those are our new invasion events. So using this kind of assay, we can have a very rigorous and quantitative ex vivo evaluation of parasite invasion into reticulocyte. This allows us to evaluate vaccine candidates, but also to look at genotype-phenotypes interaction. And so I'll just give a word about this. The kind of studies we've been doing. So when the parasite infects the red blood cell, it uses a series of receptor-ligand interactions, and this is highly regulated and very well known for falciparum, but Plasmodium vivax is the same. We just don't know which receptor and which ligand, except, and this has been described for many decades, PVDBP at the surface of the parasite and the Duffy receptor at the surface of the red blood cell, the dark antigen. And this interaction is really critical. We've known this for years. And it's, of course, then PVDBP is a major target for a candidate for vaccine development. The problem that we faced, and this is very common in malaria vaccine development, is there is a very big natural diversity of parasite, a big sequence polymorphism, and this was the main obstacle for vaccine production, and probably still is. So we try to take a different approach, going back to identifying individuals with naturally acquired immunity, getting sword B cells that were DBP specific, and check if we can detect any monoclonal antibodies that could be strain transcending, so meaning they could block the invasion of Plasmodium vivax regardless of their sequence polymorphism of their PVDBP gene. And actually this worked really nicely. We could identify a number of monoclonal anti-DBP antibodies that in our ex vivo assay were really blocking invasion into new reticulocytes and that were unaffected by the natural diversity of the parasite. So we were really happy about this. But in fact, what we realized very quickly was that a different polymorphism that we never thought would matter in vaccine development was actually driving the response of parasite against neutralization by antibodies. And this was the gene copy number, DBP being a gene, a protein that can be encoded by a gene present at high copy in the genome of the parasite. And so parasites with one copy of the PVDBP gene were neutralized by those antibodies, and the higher the number of gene copies you would have in the parasite, the less neutralized they were by the antibodies. And so this was quite unexpected, and we're currently working on trying to understand if those ex vivo results would actually translate into a failing vaccine against PVDBP. So our process allows us to evaluate new vaccine candidates. And so I told you DBP, Duffy is known for decades. And in the past 5 years, there's been a lot of renewal in the field because other groups came out with new receptor ligands that seem to be critical for interaction. And when we saw this, we wanted to evaluate in our hands using our assay how their candidates. So this is another Plasmodium vivax protein expressed on the surface of the merozoite. That it was shown to interact with the transferrin receptor at the surface of the red blood cell. Very clear and beautiful structural biology work. Where it was not that clear was if you block this interaction, do you actually block the invasion of the parasite? And what we show was that antibodies against PVRBP2B definitely recognize our parasite, no problem. They are really beautifully stained. However, they had absolutely no impact on neutralization in vitro, meaning that, yes, those RBP2B transferrin receptor interaction exist. However, it is not critical and essential for the parasite invasion. If you block these pathways, the parasite will still be able to infect the red blood cell. So in my opinion, RBP2B is a dead end for vaccine development. But we still have some good news sometimes, and recently we identified MA1 as a promising target for a blood stage vaccine. It has antibodies against MA1, so those are human monoclonals, and they have a very nice neutralizing activity in our ex vivo assay against those blood stage parasites. And really what was unexpected but really interesting is that they also block in a very nice way and at similar concentration a hepatocyte invasion of sporozoites. So this for Plasmodium vivax was the first time that we could identify a single parasite protein that was involved in the invasion of two different cell types that are critical for the parasite life cycle. So this leads me to our hepatocyte bioassay, because I think the main thing in Plasmodium vivax is the hypnozoites here, whether for elimination perspective, but also for our biological understanding of the parasite. We don't know what happens there. It's really a big black box. There's no diagnostic. Drugs that we have are toxic. There's a real need. A bit of interesting thing here. I can discuss this after. I'm pretty sure I won't have time. So how can we establish an in vitro assay for those kind of parasites? Where do you get sporozoites, and what kind of hepatocytes can sustain those infections? And so this is something we've been working a while. There's nothing magic into this. Again, it's really just about having the right logistic implemented, and where you combine parasites you get from an infected patient with mosquito colony. And what we do is after blood collection, we do a membrane feeding of our Anopheles dirus, which is the species that we have in Asia. 14 days after feeding, you get the sporozoite that you dissect from each individual female mosquito, and you infect primary human hepatocytes that were seeded in your 384 well plate 2 days before. And after X number of days, here typically 6 days, you image your plate to see, do you observe the parasite? And what we see is two different kind of forms with clearly a different size. So those huge, massive amoeboid shape forms that are the replicating schizont in the hepatocyte. So inside you have multiplication of the parasite, and then they will burst normally to enter the red blood cell. And you have those tiny forms with this typical prominence here on the side, and those are the dormant hypnozoites. So we are really happy to have this system now in the lab. We can do drug screening, we can evaluate vaccine candidates, as I showed just before, but also, and this is something that really interests me a lot, we can try to understand what's happening, what causes hypnozoite activation, for example. Could we wake up a hypnozoite and then kill it? So just, I think that's the last slide I have in terms of results, just to show one of the example of the things we can do with this liver stage assay. So we screened, so there was quite a number of mosquitoes dissected, and so this is, you have to imagine, it's each, we have 3 lab technicians sitting next to each other, dissecting under the loop individual mosquitoes to collect the sporozoites that are in the salivary gland, and then use this to infect hepatocyte. So quite a big number of mosquitoes, a huge number of sporozoites obtained. allowed us to screen more than 100,000 compounds. So this is just a reframe library. So all those drugs were discontinued or no more registered, but already approved at some point. And the idea was, can we use already approved drugs to identify anti-hypnozoic drugs? And here the very interesting thing is that we came up with a few molecules here that were showing nice activity against the parasite and not so much toxicity. And what we didn't know at that time was that actually those drugs, they target the DNA methyltransferase pathways, so epigenetic pathways that are involved in the parasite. And our question now is really, is the regulation of this dormancy in Plasmodium vivax going through epigenetic? And this was really a result of this screening effort. So yeah, to conclude, it is possible to work on Plasmodium vivax. It's quite difficult. Usually when I talk to people working on malaria and I say I work on vivax, they say, oh, vivax, no, it's too difficult, we don't work on it. There's few places in the world that can do it. We do it in Cambodia, a group does it in Thailand, there's 1 or 2 groups in South America. As long as you have a country with infected patients, you have a good logistic, and you have good lab capacity, it's something you could do. And so I was thinking, where else in the world could you have something like this? And I came up with this map where you have a very big city with amazing laboratory and logistic and infrastructure for research with infected patients that are not that far from there. And so I think that here in Korea, you could have a very nice you would have nice opportunity to establish this kind of research, and I'm pretty sure you could make great things and probably as good as what we do in Cambodia and become our competitors. So I'll finish here by saying that all this work is, of course, collaborative work. Everyone in the lab, we're a unit of nearly 30 people, and so they all contributed to this. And of course, a word on our funders. And thank you very much for your attention.